Hello everyone. In this practical we will be discussing local alignment using Smith-Waterman algorithm. In the platform, the experiment number 16 is about the local alignment of sequences. In the procedure, you can see that there is a link that is given. You can open that link. So, similar to the previous experiment, that is a Needleman-Wunsch algorithm or Emboss needle, we have a Emboss Water program is there. Emboss Water means Emboss package with Emboss package program with Smith-Waterman algorithm for the pairwise alignment of these two sequences. And keep it in mind that the previous one or the needle algorithm is for global alignment of these two sequences, whereas this one or the water algorithm is used for a local alignment of two sequences. Your sequence input may be of a protein or DNA nature. Now I am selecting the DNA itself. Okay. Okay. Now let us give two inputs. The previous one, we know that in global alignment these sequences were having only two sequences were only having 42 or 44 percentage of similarity. So now let us create another sequence from this one. For example, I am copying the sequence and pasting it in the first box. So this is the first sequence. So instead of selecting another sequence, I am selecting this region. These three lines were selected, and now I am trying to align this sequence which is of a different length. For example, a shorter sequence. Usually you know that a local alignment is done with sequences of different lengths. A smaller sequences will be locally aligned against a localized region of a larger sequence. That is how this local alignment is done. So now the same sequence, exactly same sequence is now copied and now it is pasted here. And if interested, you can say you can remove some base pairs also. So now you have a test sequence for the local alignment. So as I mentioned, Smith-Waterman algorithm or water program is used for aligning this shorter sequence with a longer sequence using the local alignment algorithm. Now we have the other options which we do not want to modify right now. We have seen that these are the options. Gap opening penalty and gap extension penalty can be changed or modified if it is required. Now we will click submit. Okay, now we have the results here. Similar to the previous example of the needle, we have the details of the program and run date and time, then different inputs that is passed into the or arguments that is passed into the command line program, etc. were given there. Now when we come to the actually aligned sequence, we have about 97.1% similarities there because it is the same sequence, and the difference is because I have deleted some of the regions of the locally aligned region. So instead of representing the entire longer sequence, only the sequences from 141st base of the longer sequence to the 350th base of the longer sequence is represented here, and both the ends of the sequence as or this region, the first three lines or the first two lines and the remaining lines after this sixth line is not represented in this alignment. Only these aligned region or the locally aligned regions of these three lines were now represented in this alignment program. So now both the sequences were represented here, and these gaps represent the bases that I have deleted previously from the second input box. So now since we have copied and pasted the same sequence from the FASTA, it is an exact match except in the region of this deletion. There is no other mutations because it is the same sequence. So this is how a local alignment is done using Smith-Waterman algorithm of the Emboss package. Thank you.